Hi everyone. Today we are deep diving into Unit 5 of APA Biology, which is heredity. This unit is all about the how and why of genetic transmission, and we're going to look at the cellular dance of meiosis, the process that ensures that you aren't just a clone of your parents, and then also explore the rules of inheritance from Mendel's pea plants to the complex non-Mendelian patterns that we see in us humans. We'll also cover the math behind the science and use chi-square to prove our theories. So let's start and let's get into it. Here are the basics of what we'll go through in this video. First, we'll go through the transmission of genetic information, then meiosis, Mendelian genetics, and non-Mendelian. Then we'll go through chromosomal inheritance, do a chi-square analysis, and then we'll talk about environmental and cytoplasmic inheritance. So first of all, to understand heredity itself, we have to understand the gear behind it. DNA is our master code, but the problem is it's packaged into these structures called chromosomes for transport and organization. Genes are specific segments of DNA that code for our traits, and most of our body cells are diploid or 2N, meaning that we have two versions of every chromosome, one maternal or from your mother and one paternal or from your father. These homologous pairs carry the same genes at the same locations, but they might have different versions, known as alleles. Gametes, your sperm and egg, are haploid, or N, carrying just one set of chromosomes so that when they meet, they can restore the diploid number. Think of meiosis as a reduction division. You replicate the DNA once during interphase, but you divide the cell twice. The goal is to turn one 2N cell into four genetically unique haploid gametes. This reduction is very important for maintaining the correct chromosome count across generations, and from an evolutionary standpoint, the fact that all eukaryotes, from yeast to humans, use these same conserved processes like spindle fibers and checkpoints is major evidence for our common ancestry. Meiosis 1 is where the heavy lifting of genetic variation happens. Specifically, in prophase 1, homologous chromosomes find each other and pair up in a process called synapsis to form a tetrad, which is shown right here. While they are physically connected, non-sister chromatids exchange DNA segments at points called chiasmata. This is crossing over. It creates recombinant chromosomes, which are totally new combinations of DNA that never existed in either parent, and this ensures that every gamete produced is a unique genetic original. Next is metaphase 1, which features independent assortment. The homologous pairs align randomly at the center of the cell, so they can either be oriented over here with pink on top and blue on bottom, or blue on top and pink at bottom. How pair number 1 lines up has no effect on pair number 2, and with 23 pairs in humans, this generates millions of possible combinations. Then, in anaphase 1, these homologous chromosomes are pulled to opposite poles, and this specific movement, the cell transitions from diploid to haploid, even though the chromosomes are still in the duplicated sister chromatid format. After that specific movement, the cell then goes through telophase 1 and then cytokinesis, which will form two genetically unique cells. After meiosis 1, the cell moves directly into meiosis 2 without replicating its DNA again. This stage looks exactly like mitosis. We line the chromosomes up at the equator and then pull the sister chromatids apart during anaphase 2. By the end of telophase 2, we have four genetically unique haploid cells. Remember, meiosis 1 separated homologous pairs, and meiosis 2 separates the sister chromatids. It is easy to confuse mitosis and meiosis, so let's be clear. Mitosis is for growth and tissue repair, making two identical diploid cells. Meiosis is strictly for sexual reproduction, and it makes four unique haploid gametes. While mitosis is one and done for division, meiosis goes through two full rounds to ensure maximum genetic variation and the necessary reduction in chromosome number. Now, let's transition from mitosis and meiosis directly into the actual laws for inheritance. Gregor Mendel's work gave us the rulebook for inheritance itself. His first law, law of segregation, states that two alleles for a trait separate during gamete formation, so each gamete gets only one. His law of independent assortment says that genes for different traits on different chromosomes settle into gametes independently. Lastly, his law of dominance explains how dominant alleles can mask recessive ones and show up in a heterozygote. Now, let's transition from Mendel's laws and see how they actually apply in the real world. We use Punnett squares to predict these genetic odds. A monohybrid cross between two heterozygotes, let's use this example up here, which is big P and little p, will typically yield a 3:1 phenotypic ratio, which means you will see the dominant allele appear 3 times, where you'll see the recessive allele clearly appear only once. A dihybrid cross, on the other hand, for two traits, let's use big Y little y and big R small r, will yield a classic 9:3 to 3:1 ratio. If you ever encounter an individual with a dominant trait but you don't know their genotype, you will perform a test cross by breeding them with a homozygous recessive. Let's use the example up here. So instead of big P, little p, you will use two small p's right here. And you will breed this homozygous recessive individual with them to see if any recessive offspring appear. Now, let's talk about the chi-square analysis. Genetics itself is a numbers game, and we use the chi-square test to see if our real-world observed data matches our Mendelian expected ratios. We calculate a value based on the difference between the two. If our calculated chi-square is lower than the critical value from the statistic table, we fail to reject the null hypothesis, meaning that data fits our model and Mendelian ratios are at play. But if it's higher, it suggests that something else is at play, like gene linkage or non-Mendelian inheritance. Now, what is non-Mendelian inheritance? So Mendel, in his initial tests, used P's, which were very simple, but life is often very complex, on the other hand. We have incomplete dominance, where phenotypes blend like pink flowers, and codominance, where both traits show up at once, like AB blood types. Multiple alleles mean more than two options exist in a population, while polygenic inheritance means many genes control one trait, like your height or skin color, for example. There's also epistasis, which means one gene can essentially override or mask another gene's expression. Additionally, your DNA isn't the only thing in charge of your looks. Phenotypic plasticity is the ability of one genotype to produce different phenotypes depending on the environment. A classic example is a hydrangea plant. The same genetic individual will produce blue flowers in acidic soil, but pink flowers in alkaline soil. The genes remain the same, but the environmental pH can change how these genes are expressed and produce a different phenotype depending on the soil quality. Additionally, not all of your DNA is in the nucleus itself. Mitochondria and chloroplasts contain their own circular DNA. In humans, you get all of your mitochondria from your mother's egg cell, not the sperm. This is known as maternal inheritance, or even mitochondrial or cytoplasmic inheritance. This means that if a mother has a mitochondrial or cytoplasmic trait or disorder, she will pass it to all of her children, while an affected father will pass it to none of them. Now, let's use all of this information and put it into a pedigree. Pedigrees are family trees that track specific traits. We use them as logic puzzles. If a trait skips a generation, it is likely recessive. If it appears in every generation and affected children always have an affected parent, it's likely dominant. If it affects males much more frequently than females, we suspect an X-linked recessive trait. And mastering these patterns is a huge part of the AP exam and even just the Unit 5 heredity test. Finally, let's look at the physical chromosome behavior. Linked genes are located so close together on the same chromosome that they rarely cross over and usually inherit as a unit. We also have sex-linked traits governed by the X or Y chromosomes. Sometimes chromosomes fail to separate correctly during meiosis. This is called non-disjunction. This results in gametes with an incorrect number of chromosomes, which can lead to conditions like Down syndrome and also lead to no more cell division, which means the cell will go into the phase G0 or just perform apoptosis. Now that we're done with all of our review, let's put this into practice. Looking at this pedigree, is the trait dominant or recessive? Also, is the trait likely to be sex-linked or not sex-linked? Here's a hint. There's an equal split between males and females, and the trait also appears in every generation. So please pause the video here and come back when you have an answer in mind. So the trait is likely to be dominant because every generation contains it, and for each affected child, there's at least one affected parent that goes along. The trait is also likely non-sex linked or autosomal because there's an equal male to female inheritance ratio. For 3 females, we can also see 3 males inherited it. Now, let's move on to question 2. Please read this on your own, look at the information provided in the graph, and then come back when you have an answer in mind. So, the correct answer choice is option A. The chi-square value is 1.53 and the null hypothesis cannot be rejected. So first, after we do our calculation, the value that we get for chi-square is 1.53. And this value is much less than the critical value of 5.99, which is provided right over here. So because the calculated value is lower than the critical value, the null hypothesis cannot be rejected, and this means that the observed data fits the expected model of incomplete dominance. Here are some key takeaways for this video. Let's wrap up. Meiosis drives diversity through crossing over and independent assortment. Mendel gave us the foundations of inheritance, but things like linkage, organelle DNA, and environment adds additional layers of complexity. We use statistical tools like chi-square to verify our findings, and ultimately these shared genetics processes across all life highlight our common evolutionary history. Thank you for joining me in this Unit 5 review. Keep practicing those pedigrees and chi-square problems, and I'll see you in the next unit.